Okay, so muscle contraction. Again, you guys are going to have to bear with me. This is the first time I'm seeing this lecture in about a week or two, so I have to deal with my my mom brain, my momnesia. Okay, if we can get the first slide to advance, we'd be in good shape. Or not. There we go. Alright, before we get into the contraction, I just want to cover a couple more muscles on the anterior abdomen. Okay, we're looking at the six-pack rectus abdominis. Okay, rectus abdominis is what most people know as their six-pack. It's the muscle in the center of the anterior abdomen. The other muscles we're going to look at are the obliques, the external abdominal oblique, the internal abdominal oblique, and transverse abdominis. So, rectus abdominis first. Rectus abdominis has parts of its own, namely linea alba. That literally means white line. It's the line of connective tissue of fascia running directly down the center of rectus abdominis, dividing it into two halves. Those two halves have tendinous intersections. There's one at the level of the xiphoid process, one at the level of the umbilical cord, and then one halfway in between. So that's how we're getting the six-pack look. You have three sets of two muscles on each side. Okay? Linea semiluminaris, okay, that's a half circle, is to either side framing or outlining both sides of rectus abdominis. So linea alba straight down the middle, linea semiluminaris to either side. The rectus fascial sheath completely encloses rectus abdominis. So if we were to take off your skin, take off the greater omentum, and then we were to look at your anterior abdomen, this is not what we would see. We would see the rectus fascial sheath, a large piece of connective tissue covering those muscles. If we cut the rectus fascial sheath, then we would in fact see the muscle rectus abdominis. If we look at the obliques, you have two sets, the external abdominal oblique and the internal abdominal oblique, thusly named because the external is more external and the internal is more internal. Obliques are going to be to the sides, and this works out the same way as the intercostals. Remember that external intercostals between the ribs ran front pocket and internal intercostals ran back pocket? Same thing applies with the obliques. The external abdominal obliques run front pocket. The internal abdominal obliques run back pocket. Okay? In order to see the internal abdominal oblique, you must first remove the external abdominal oblique. Internal is deep. The deepest of all three is called transverse abdominis, named because the muscle fibers run transversely. If we were to cut the body according to the way these muscle fibers run, we'd be taking a transverse section. So, in order from superficial to deep, when you're looking at the side of the abdomen, it goes external abdominal oblique, then internal abdominal oblique, then transverse abdominis. The inguinal ligament, we're revisiting it because remember we said it is comprised of tendons and fascia coming from the abdominal muscles. From both rectus abdominis and the oblique muscles, they all contribute to the inguinal ligament. Now the inguinal ligament is generally more pronounced in males and of more importance in males because it can be a spot where hernias can occur. Okay, now we're going to get our first taste of physiology. Okay, muscle anatomy and the development. Some of this we visited in histology. So a single skeletal muscle cell is a muscle fiber. Okay, when you are an embryo, several single cells called myoblasts will fuse together into a cylindrical muscle fiber. That's why one muscle fiber has many, many nuclei, because originally it was the combination of many, many cells. Now, after you are born, no new muscle fibers are ever formed from myoblasts, but some mild regeneration can occur by a cell type called a satellite cell. Okay, so damaging muscle. When you damage your muscle or you lose muscle fibers, some muscle will be regenerated by satellite cells, but it's pretty nominal. It's not a lot. But what happens is if you lose 50%, let's say, of your deltoid muscle, will you lose 50% of the function? And the answer is no, because of hypertrophy. Hypertrophy, this is revisiting this word, because when we talked about what it means to be alive and a living organism, we talked about hypertrophy and hyperplasia. Hypertrophy is when the remaining muscle fibers enlarge themselves. So again, if we go back to thinking about losing 50% of your deltoid muscle, do you lose 50% of the function? The answer is no, because the muscle fibers that are left are going to enlarge and take over for the function of the ones that you lost. Okay. So this is our overview picture. This is where we're going to get down to the molecular level. This is what we've been doing, the gross anatomy. We're looking at a muscle fiber. We're going to start to pull that muscle fiber apart into the myofibrils and down to the sarcomeres, the actin, and the myosin. Okay? We know that muscles are linked to bones by tendons, and we know that the striation that appears in skeletal muscle is due all the way down on the molecular level to thick and thin filaments called myofibrils. Okay, each muscle fiber is filled with myofibrils, and it goes from one end of the muscle to the other. Myofibrils are made up of sarcomeres, and sarcomeres are made up of thin and thick filaments. The thick filament is myosin, and you have three thin filaments: actin, troponin, and tropomyosin. Okay, just to give you an idea where we're going to go with this, what I do anyways, when I talk about physiology, we're going to start with the muscle contraction, and then we're going to work our way backwards. We're going to say, all right, the muscle contracts. What had to happen in order for that muscle to contract? And what we'll end up with by the end of the lecture is an ordered sequence of events that have to happen in order for a muscle to contract. And you're going to want to know that ordered sequence of events. You're going to want to be able to repeat it to yourself without looking at the notes, without looking at the textbook, without listening to this lecture. You're going to want to get that sequence of events in your head. Okay, so we'll start with the muscle contraction itself. How do we get that? In a sarcomere, you have spaces between the thick and thin filaments. Okay? The thick and thin filaments are able to touch each other through a projection called a cross bridge. The cross bridge is a structure from myosin, the thick filament. So myosin molecules have a projection that extends from the thick filament and can go over and touch the thin filaments. During a muscle contraction, those cross bridges, when they touch each other, is what allows force to be exerted and the muscle to contract. So here we are down where the green box is. Here's your sarcomere. And when we look at the thick filament, we see those projections. When we put it into a sarcomere, you can see those projections are reaching out towards the thin filaments here in purple. When they make contact, they exert force on each other, and that's what allows a muscle contraction to occur. Okay, first thing you want to get in your head is the problem with the word shortening. Okay, muscle contraction does not necessarily mean shortening. When we talk about muscle contraction, we're talking about generating force within muscle fibers, and the site of that force generation are the cross bridges, the small projections off of the thick filament. When the thick and thin filaments move across each other, okay, they don't change length. Myosin never gets any shorter, actin never gets any shorter, okay, but they are sliding past one another. This is called the sliding filament mechanism of muscle contraction. Okay. Muscle contraction does have shortening in it somewhere though, okay? The common pattern of muscle shortening where one part remains fixed and the other position moves towards it. And when we look at a sarcomere, this will make more sense. Okay, here we're looking at two sarcomeres. One is relaxed, one is contracted or shortened. Okay, I'm not requiring you to know the anatomy of a sarcomere. So you'll see all of these things, A band, I bands, H zones, okay, Z lines. I'm not going to ask you those on the test, but they are useful for looking at the difference between a relaxed and a contracted sarcomere. In a relaxed sarcomere, notice that there's a large gap between the myosin, the end of myosin, and the end of actin, and there is a big gap between these two actin filaments, or these two thin filaments. In a contracted muscle, the cross bridges have attached to the actin, moved across, and they have shortened those distances. Okay, this gap between the end of myosin and actin is gone. The gap between these two thin filaments is shortened. Okay, that's where you're getting shortening. But as a whole, this thick filament never changed length. These thin filaments never changed length. So be very, very careful when you think and use the word shortening when you're talking about muscle contraction. Okay, these next set of slides actually came from your book. You can use them to click through a muscle contraction. It will take you step by step, and at the bottom of each slide it will tell you what's happening. So this is just an overview. They put all those different zones in different colors. The green is myosin, or your thick filament. The purple is actin, troponin, and tropomyosin, which are your thin filaments. And then here's a fully contracted muscle. Notice those gaps have gone away. So if we look, here's the first one, just giving you the anatomy of the sarcomere, completely relaxed. Then as it contracts, myosin grabs onto actin and moves, generates a force, slides across it. Okay, and this gap is starting to shorten, and this gap is going to start to shorten. Happening even more, and you have a fully contracted muscle. Okay, the H zone is gone, the I band narrows, but the A band, which is the length of actin and myosin, are unchanged. So again, be careful with the shortening. Okay, actin itself. Actin is a globular protein, okay, and it is a single polypeptide that polymerizes with other actins to form two intertwined helical chains. So basically, when we look down here, it looks like two strings of pearls twisted around each other. Globular means each one of the pearls is a globular protein unit. Okay, it polymerizes, so many units, polymer. Okay, many units together, the same unit repeating. You form two what look like strings of pearls. You twist them around each other. Then you've got actin. Each actin molecule, each one of these globular proteins, you see a dark space under there? That's one binding site for myosin. Okay, looking at myosin. Myosin is two large polypeptide heavy chains and four smaller light chains. When all of these chains come together, you get a molecule that has two globular heads, which are your cross bridges, and a long tail. The tail of each myosin molecule is going to lie along the axis of the thick filament, which means that basically means all the tails run the same direction. And the two globular heads are what form your cross bridges. What you'll notice about one myosin molecule is that it has two distinct ends. Okay, each end has heads or cross bridges that are oriented in opposite directions, and then the tails go towards the center of the thick filament. So here's a picture. Two globular heads, two heavy chains, four light chains. The two heavy chains twirl around each other. The two heads, the globular heads, have actin binding sites, and they are what form the cross bridges. Notice all the tails, all the tails lie along the same axis, and that we could divide it right down the middle and see that there are two distinct ends where the heads are oriented in opposite directions. Okay, here starts the very beginning of our sequence of events. So far we've just been talking in generalities about muscle contraction, but now we're going to start to talk specifically about what needs to happen in order for a muscle contraction to occur. Alright, 4 parts to what's called the cross-bridge cycle. The cross-bridge cycle is the sequence of events that occurs between the time the cross-bridge, the projection on myosin, reaches out, binds to the thin filament, and then moves and repeats the process. So the first step, attachment of the cross bridge to a thin filament. The head of myosin, those globular heads, reach out to actin and attach themselves. Step 1. Step 2 is the movement of those heads, walking themselves along the actin filament, okay, shortening the sarcomere. The third step is to detach. And the fourth set is to energize, to repeat. Okay, they need more ATP. You can liken this to rowing a boat. Okay, this works out much better when I'm sitting in class and I can show you, but let's see if we can do this. If you are sitting in a boat, okay, the boat's in the water, you and the boat are myosin. You're the thick filament. You've got 2 oars. Those are your globular heads, okay? You are going to put your oars in the water. That's the first step, attachment. The water is actin, you and the boat are myosin. You put your globular heads, your oars, in the water. Then you pull your oars towards you, okay? That propels the boat through the water. That's the movement. Then you pick your oars up out of the water. That's detachment. And then you reset to your original position and you get ready to take another stroke through the water. That's step 4. Energizing, repeating, resetting. Okay, so you're myosin, the water is actin, the oars are the globular heads. You attach, you put your oars in the water, okay, that's the globular heads attaching to actin. Then you pull the oars toward you, which propels the boat through the water, that's the movement. Then you pick the oars up out of the water, that's the detachment, and then you reset and re-energize. Okay, so that's the cross bridge cycle. Now we're going to back up another step, and we're going to say what has to happen in order for the cross bridge cycle to go forward. And before we can talk about that, we have to talk about the other thin filaments. Remember that myosin is the thick filament, but you do have 3 thin filaments, troponin, tropomyosin, and actin. So let's look at troponin and tropomyosin. Tropomyosin, okay, this is just a description, it's a rod-shaped molecule, it's also 2 intertwined polypeptides, and 1 tropomyosin is equal to 7 actin molecules. Tropomyosin covers the myosin binding site on actin. So when tropomyosin is in effect, myosin and actin cannot bind to one another. Tropomyosin blocks that binding. Tropomyosin is held in place by another protein called troponin. So here's troponin, the pink one. Here's tropomyosin, the blue one. So if we count, we've got 1 blue tropomyosin molecule. 1, 2, 3, 4, 5, 6, 7. It's in contact with 7 actin molecules. It is blocking, in its current condition, it is blocking the binding sites on actin for myosin. Troponin is holding tropomyosin in place. Okay, so troponin is the boss of tropomyosin. Troponin can interact with both actin and myosin. Okay, the small pink protein troponin has 3 subunits, an I, a T, and a C. The I is inhibitory, the T is what allows it to bind to tropomyosin, and the C allows it to bind to calcium. 1 molecule of troponin binds to each molecule of tropomyosin and regulates access to 7 actin molecules. So you can say that 1 troponin can block or allow 7 actin molecules to interact with myosin. Okay, we got to bring calcium into this. Calcium is king of muscle contraction. And remember, we're backing up. We're saying what has to happen for those 4 cross-bridge steps to occur. Calcium binds to troponin on the calcium binding subunit. That's the only thing calcium binds to. It does not affect tropomyosin directly. It does not affect actin directly. Binding of calcium to troponin causes a conformational change. So essentially, when calcium binds to troponin, it twists troponin. And twisting troponin, since it's attached to tropomyosin, twists tropomyosin. And if you twist tropomyosin, you pull it off of the binding sites, and now myosin and actin can bind to each other. So calcium has to bind troponin. It causes a conformational change in troponin, which causes a conformational change in tropomyosin, which then reveals the myosin binding sites on actin, and then you can start your cross-bridge cycle. This continues until calcium is removed from troponin. So here, if we take a look, here is a relaxed muscle. There's low calcium around, okay, so what we see is troponin, tropomyosin are helping to block the binding sites on actin. Myosin cannot come across and form the cross bridge. Once cytosolic calcium is high, calcium binds troponin, causes a conformational change in troponin, which causes a change in shape of tropomyosin, pulling it off of those myosin binding sites on actin. Now the cross-bridge cycle can begin. So if we just stop at this slide for a minute and we start to say our sequence of events, starting with calcium. Calcium comes near the muscle fibers, it binds to troponin, it causes a conformational change in troponin, which causes a conformational change in tropomyosin, which pulls tropomyosin off of the myosin binding sites on actin and allows the cross-bridge cycle to go forward with the 4 steps, which are attachment, movement, detachment, reset, and re-energize. So up until this point, you want to make sure you can say that whole sequence of events to yourself. Okay, how do we get the calcium? We're backing up another step. Okay, remember we started with muscle contraction, then the cross-bridge cycle. Then we said, what do we have to do to get the cross-bridge cycle? Well, it all started with calcium. Well, what do we have to do to get the calcium? So we're backing up even farther. In order to get the calcium, we have to let the muscle fiber go through something called excitation-contraction coupling. Excitation will almost always refer to something having to do with a nerve cell. Contraction is our muscle. So excitation-contraction coupling has to do with the sequence of events by which a nervous signal, which is called an action potential, comes to the plasma membrane of a muscle fiber and ultimately leads to the cross-bridge activity. Okay, why do I feel like I skipped one? Nope. Okay, something that's interesting about excitation-contraction coupling, the action potential, and this is the signal coming from your brain going to your muscle, lasts 1 to 2 milliseconds. Now I know we don't deal in milliseconds, it doesn't mean a lot to you, it's about 1/1000th of a second. Okay, it's very quick. And the signal for your muscle to contract is over way before the muscle actually contracts. What's interesting is the mechanical activity, the actual contraction, lasts 100 milliseconds or more. Again, we're still in milliseconds, but what you can realize is that contraction actually takes 100 times longer to achieve than the signal that was sent to cause the contraction. The electrical activity, that's the signal coming from the nerve, in the plasma membrane does not directly act on those contractile proteins. It's a way of providing calcium, and that should make sense because we know it's all about the calcium. Okay, in a resting muscle fiber, the concentration of calcium is very, very low, 10 to the -7th. Okay, that's 0.6 zeros, then a 1, very low. So therefore, not a lot of troponin has calcium on it. So if troponin does not have calcium, that means tropomyosin is blocking myosin and actin from binding to each other. Following an action potential, following a signal from your brain to your muscles, there's a rapid increase in calcium. Calcium binds to troponin, causes a conformational change, causing a conformational change in tropomyosin, which pulls it off of the myosin binding sites on actin so that the cross-bridge cycle can begin with attachment, movement, detachment, reset, re-energize. Okay? That's exactly what you need to do. Anytime you get a chance, say the sequence of events. The source, where did that calcium come from? It came from something called the sarcoplasmic reticulum. The sarcoplasmic reticulum should almost sound familiar because it sounds a lot like endoplasmic reticulum. It is a homologous structure, which means it's very, very similar, okay, but it's been altered in muscle cells into something called the sarcoplasmic reticulum. Sarcoplasmic reticulum is a structure that forms a sleeve around each myofibril. So what we're looking at here, the gray is the plasma membrane of a muscle fiber. Inside of a muscle fiber, we have the myofibrils. This blue Swiss cheese looking sleeve is the sarcoplasmic reticulum. That's where all of the calcium is stored. At the end of each segment of sarcoplasmic reticulum are 2 enlarged regions called lateral sacs. That's where the calcium is stored. There's another structure inside the cell called a transverse tubule, or a T-tubule. T-tubules associate with the plasma membrane and the lateral sacs. Okay, that becomes important because that's the messenger from the plasma membrane to the sarcoplasmic reticulum. The mechanism that couples the two, we're not going to talk about. All you have to know is it's the messenger. So the action potential comes down to the plasma membrane. The T-tubule picks it up from the plasma membrane, carries it to the lateral sac of the sarcoplasmic reticulum, and then we get calcium release. So if we look at the pictorial representation, remember this gray is the plasma membrane of a muscle fiber. This is where a nerve would come down and touch the plasma membrane. Nerves cannot go inside of the muscle fiber. They can only touch the membrane. So the nerve brings its message down. That message is taken from the plasma membrane by the T-tubule and transferred to the lateral sac. So here's one lateral sac. Remember, it's at each end. Here's another lateral sac. That's where all the calcium is stored. Once the calcium is released, you can see how these myofibrils would be completely bathed in calcium. Okay, so let's just say everything again from beginning to end. Well, it doesn't really apply to this slide, but we're just taking some time to say it. So, first thing that has to happen for your muscle to contract is your brain has to decide it wants it to contract. So an action potential comes down from a nerve cell to the plasma membrane of a muscle cell. The action potential is received by the plasma membrane. It's picked up by the T-tubule. The T-tubule passes the message on to the lateral sac of the sarcoplasmic reticulum. Calcium is released. Calcium is then free to bind troponin, causing a conformational change in shape. Okay, change in shape in troponin causes a change in shape in tropomyosin. Changing the shape of tropomyosin pulls it off of the myosin binding sites on actin, allowing the globular heads, the cross bridges of myosin, to bind, do the cross bridge cycle, which is 4 steps: attachment, movement, detachment, reset, re-energize. This all will continue until calcium is removed. So those are all the steps from beginning to end for a muscle contraction. Okay. Calcium release, once it's released, it's usually one action potential, one signal from the nerve can saturate all the troponin binding sites on the thin filaments. Okay, so it's one signal is usually all you need to get your muscles to contract. How do we get the muscle to relax, returning to rest? And this falls under the part where we talked about the signal only taking 1 millisecond, but the contraction taking 100 times longer. This is why. When you need to get rid of the calcium, it has to be pumped back into the sarcoplasmic reticulum. Here's the problem. It was high in the sarcoplasmic reticulum and low out in the muscle fiber. So if you want to go the opposite direction, you're trying to move calcium from an area of low concentration to high concentration. Calcium does not want to go that way. Okay, that requires active transport. So ATP is going to be needed to pump it back into the sarcoplasmic reticulum. This makes calcium very unhappy, and this is why cytosolic calcium concentration can remain high for a longer period of time after a single action potential, because it takes a long time to take the calcium and force it back into the sarcoplasmic reticulum. It does not want to go that way. Okay, exciting the membranes on the muscle fibers. So when a nerve fiber goes to a skeletal muscle, this is the only way action potentials can be initiated in a skeletal muscle. Okay, that means there's only one way to make a muscle contract, and that's for your brain to say so. That's a skeletal muscle, by the way. Okay, that's the only type of muscle we're talking about. So that first bullet point says the only way you can make a muscle contract is through the action potential of a nerve. That means your brain decides to. The nerve cell whose axons come down and innervate or touch a muscle fiber are called motor neurons. Makes sense because we're talking about skeletal muscle. That's motor, that's movement. So the nerve responsible for contracting the skeletal muscle is a motor neuron. The cell bodies are in the brain stem or spinal cord, and they have a very large diameter axon, and they can propagate, which means they can move their signal, their action potentials, very, very quickly. Okay, when a motor neuron touches a muscle fiber, that's called neuromuscular junction. one motor neuron can touch many, many muscle fibers. Okay, so what we're looking at is the cell body right here. That's what would be in the brainstem or spinal cord. This is the axon. Okay, it's transmitting information, and then it branches off and can touch many, many muscle fibers. One motor neuron plus all the muscle fibers it touches is called a motor unit. So what you're looking at, this whole picture, is a motor unit. When an action potential occurs in a motor neuron, it affects all the muscle fibers it touches. That one motor neuron cannot be selective. It can't say, even though I'm touching 10 muscle fibers, I only want 5 of them to contract. It doesn't work that way. Once that motor neuron gets the signal, all the muscle fibers it touches are going to contract. And here is a scanning electron micrograph of a motor neuron coming down. And we see these 3 muscle fibers. We know that they are muscle fibers because we see the striation, and we see that, again, the muscle fiber can only touch the plasma membrane. It cannot go, or the neuron can only touch the plasma membrane. It can't go any deeper. And then here's an artist rendering of the same thing, the terminal end coming down, touching the plasma membrane, and then we have the sarcomeres down here. Okay, not the best job I've ever done on muscle contraction. It's much easier for me to sit in front of you so I can make hand gestures and draw on the board, but I hope that it did help you guys.